Okay guys, hello. Today we're going to talk about DNA, or DNA is actually the first letter of one big word. It's deoxyribonucleic acid, and RNA is ribonucleic acid. This molecule, DNA, is a macromolecule, like proteins, polysaccharides, and it is also a polymer. That it is made up of many similar, smaller molecules joined into a long chain. These small molecules are called nucleotides, both in DNA and RNA. But therefore, DNA and RNA are called polynucleotides. Okay, both DNA and RNA are polynucleotides. Now, they are often referred to simply as nucleic acids, both DNA and RNA. The DNA is made up of two chains, as you can see here, one, and this is the second one, made up of nucleotides. Now we're going to look at the structure of nucleotides. Nucleotides are made up of three smaller components: a nitrogen-containing base, a pentose sugar, we will see what pentose mean, and a phosphate group. Let's start with the nitrogen-containing bases. First of all, nucleotides, we said that they make up both DNA and RNA, but there is one small difference. Deoxyribonucleotides make up DNA and ribonucleotides make RNA. Okay, and we will see where is that difference. It's not on the phosphate, it's not on the base, it's actually on the base and on the deoxyribose sugar. First of all, let's start with the nitrogen-containing bases. There are 5 of them, found in both DNA and RNA. In DNA, there can be 4. In a DNA molecule, one can find 4 of them. 4 types of them. Okay. And in an RNA, also 4. Now, let's see. There is adenine, thymine, guanine, and cytosine. Cytosine. Okay. In an RNA molecule, there are 4 adenine, but instead of thymine, there is uracil, I don't know how it's called, guanine and cytosine again. I'm going to call them by the first letter A, T, G, C, and A, U, G, C again. There has been a typo here. Okay. Now, these nitrogen-containing bases I don't have their structure in a photo. That seems highly unlikely. Anyway, here, this is a nitrogen-containing base. Not even here. Just give me a second, guys. I'm sorry about that. No, I don't. Anyway, this is a nitrogen-containing base. It has nitrogen, and in every corner, I don't know if you know that, if you have learned this, in every corner there are carbon atoms. And we number this because to know about which carbon atom we're talking each time. And the carbon atoms that are in the nitrogen-containing bases, we number them with numbers 1, 2, 3. That means, I will give you an example now. I don't know which. Let's see, the C4 is attached here to the sugar, which means the carbon atom number 4. That would be 1, 2, 3, the 4th here. I don't know where does it start, its numbering, but I want you to know that the numbers that we number the carbon atoms with 1, 2, 3, 4, etc. Now let's see the pentose sugar. It's a sugar which has 5 carbons. There are 2 types of sugars. Okay. It can be ribose in RNA or deoxyribose in DNA. As their name suggests, deoxyribose is almost the same as ribose, but it has 1 fewer oxygen atoms in its molecule. You can see that here. This carbon atom has one hydrogen and one hydrogen attached to it on the deoxyribose. But on the ribose, it has one hydrogen and one hydroxyl group. That means ribose has one more oxygen atom than deoxyribose. This is the second difference. So if we were to find differences between the nucleotides of DNA and the nucleotides of RNA, the first thing is the sugar. Is it in DNA? It's deoxyribose, one fewer atom, oxygen atom, and ribose. The second change is thymine and uracil. Okay. Now, the carbon atoms here must be numbered as well. But if we say the carbon 1, the carbon 2, the carbon 3, 4, and 5, we can separate them by these carbon atoms. So if I tell you the carbon 3, the C3 for instance, which one? These three or this one? You wouldn't know. That's why we put this little accent. I don't know how to call it, accent here. Okay, this is how we call it in Greek, so I just guess it is accent. So we've got the C1 accent, the 2 accent C, the 3 accent C, the 4 accent C, and the 5 accent C. Here, in the 5 accent C, in this carbon atom, in the 5 accent carbon, it's attached the phosphate group, while the nitrogen-containing base is attached to the C1 accent. You can see that the C3 accent has a hydroxyl in both of them attached to it. Now, we said that DNA is a polymer. That means that we have smaller molecules, nucleotides, that are joined into a long chain. That means that these smaller molecules need to be bonded together. How are they bonded? They create this phosphodiester bond. It's a bond between the nucleotides. And where it happens, you see, this is a nucleotide. It happens between the hydroxyl that is attached to the 3 accent carbon of the first nucleotide, of the sugar of the first nucleotide, with the phosphate group which is attached to the 5 accent carbon of the next nucleotide. And this is called a phosphodiester bond. And because it is between the 3 accent carbon and the 5 accent carbon, it's called 3 accent, 5 accent phosphodiester bond. Okay, now you see, in its DNA molecule, because of this type of bonding, the first nucleotide will have, you see why? This is the first nucleotide, okay? If we attach it with many others, it will still have the phosphate group free, because it's at the beginning. It doesn't have a nucleotide above it to bond with, so it has a free nucleotide group. The same happens with the last one. It has a free hydroxyl group. You see, no matter how many nucleotides are in a chain of DNA, in a polynucleotide chain, always the first nucleotide will have what? This is wrong actually. I'm going to, okay. Well, which will have what? The 5 in a phosphate group, the first nucleotide will have a phosphate group attached to it, 5 carbon atom, and the last one will have a hydroxyl group, a free hydroxyl group attached to the 3 accent carbon. So we say that the DNA strand has an orientation from 5 accent to 3 accent. This is how it goes. This arrow shouldn't be like that. Actually, yes, this is wrong. It should be like that. It goes from the 5 accent to the 3 accent. Same in here. Okay. And also we say that the last nucleotide, which has the 3 accent the hydrogen group attached to the 3-accent carbon is the 3 end of the chain, and the first one, which has the free phosphate group attached to its 5-accent carbon, is the 5-accent end. Okay. Now, we're going to talk about the structure of DNA. We said the DNA is made up of 2 chains. Okay, we saw how each chain is made, but how these chains are combined in order to create this molecule, the DNA. In 1953, Watson and Crick, 2 scientists, discovered the structure of DNA, one of the biggest accomplishments of their era in the history, actually, of molecular biology. And they took, both of them, a Nobel Prize in 1962. Let's see now what this model tells us. I'm going to look at this picture. First of all, it talks about that. How are the 2 chains together? It says that DNA consists of a double helix. That's how we say that, because there are 2 chains that revolve around each other, you see, and it's right-handed double helix, 2 right-handed polynucleotide chains that are coiled about the same axis. Now, each chain, now I have uncoiled the chain here in order to look at it, is made up of what? You see that it has a backbone. That means something steady. What is that? Sugar, phosphate group. No, sugar, phosphate group, sugar, phosphate group, sugar, phosphate group, sugar, phosphate group. And they are attached by what? These together? By phosphodiester bond. Okay. This backbone is steady and it is hydrophilic. So it is on the outside of the molecule. On the inside of this steady backbone, there are the nitrogen-containing bases, which are hydrophobic. So here you see this backbone, and these are the bases, the nitrogen-containing bases. Okay. Now, the bases of the first strand with the base of the second strand, they create between them hydrogen bonds, and this is why the 2 chains are stay together, because of these hydrogen bonds. But hydrogen bond happens only between certain bases. Adenine always pairs with thymine or uracil if we are in RNA, but now we're talking about DNA, so adenine always pairs with thymine, while guanine, G, always pairs with C. The hydrogen bonds, they stabilize the secondary structure of the molecule. Okay. Now, between adenine and thymine, there are 2 hydrogen bonds, and between guanine and cytosine, between G and C, there are 3 hydrogen bonds. So, I don't think there is ever going to ask that of you. If they ask you how many bonds are in a molecule, in a DNA molecule, this is what you have to do. How many A's you have, how many adenines? 2 times 2 plus 3 times G, which G is the number of guanines. Because you see, if there is 1 guanine, there are 3 hydrogen bonds. And if there's 1 adenine, there are 2 hydrogen bonds. If there is a second adenine, there are 2 more hydrogen bonds. And there is 1 more guanine here, or this is a mistake, okay? There are 3 hydrogen bonds here. Okay, so you get the picture. This is the formula you should use. Alright, now let's move on. Now, DNA, how it exists in the nucleus? Is it in this form, a long chain? No, actually in every nucleus there are many DNA molecules. This is important. For instance, in the human, there are 46 long DNA chains. The number is specific to the species we're talking about. And there is a specific number of DNA molecules, of chains in each nucleus. But now we're going to see how each one of them exists, in what form. So first, the DNA is wrapped. Let me show you this here. We've got this, the DNA, okay, double helix. It is being wrapped around this. You know what this is? There are 1, 2, 3, 4, 1, 2, 3, 4 below that, 8 in total histone molecules. Histone is a protein. DNA revolves around that. And then one more histone molecule, which is called H1 histone, comes above it to stabilize it. You see that between nucleosomes there is a DNA, a part of DNA which is called linked DNA, which is 53 base pairs. Okay. Okay. Now, so we've got this structure is called a nucleosome. Now, nucleosomes fold up. They fold up. They together, they coil even more. And we have these larger and larger structures until they make that. This here is called a chromatin fiber. So you see, when DNA coils, there is chromatin fiber. What we see in a nucleus is this, the chromatin. And you will be like right now, what? Chromatin. Chromatin is what? The combination of DNA and proteins. Okay. Chromatin exists in two forms, euchromatin and heterochromatin. The euchromatin is loosely coiled, while the heterochromatin is tightly coiled. You see, when something is really coiled, nothing can happen in there, because proteins, enzymes, cannot read this part of the molecule because it is really tightly coiled. This is why this type of DNA, heterochromatin, is inactive, while euchromatin, which is loosely coiled, is active. And you see, the more tight something is, the more dense it is. Therefore, in the electron microscope, we can see this is heterochromatin and this is euchromatin. The white is euchromatin and the black is heterochromatin. Okay. Now, let me look at the cell cycle. What is that? It's the sequence of events that takes place between one division to the next. But let me be even more precise to you. It's what happens at every moment in the cell. Every cell is in one phase of the cell cycle. So we can say about that phase what happens. And let me show you the cell cycle. This is one. It consists of the interphase and the mitosis. And the cell division, actually. Let's call it that. The cell division. I won't look into it right now. I will look into that, the interphase. The interphase consists of three phases, G1, S, and G2. During G1, the cell makes RNA, enzymes, and other proteins needed for growth. Okay, it carries out its normal functions. It does what a cell does. After G1, here, there is a checkpoint. There is one moment here that the cell decides, should I divide or should I not? If it decides to divide, then it goes into the S phase. If it does not, it stays in the G1. The S phase is what? In order for a cell to divide and to have two identical daughter cells, its daughter cells would have the same amount of DNA, right? But how if I have N let's say I have n amount of DNA and I want to give its daughter nuclei, its daughter cell actually, n. Is that possible? No. First, my n, I need to double it to make it 2n, and then I have to divide and give n and n to its daughter cell. And that's what happens during the S phase. It doubles its DNA, it synthesizes DNA, it replicates the DNA in the nucleus. Then, during G2, the cell is prepared for cell division. That means it creates the proteins needed for cell division and it corrects any mistakes that have happened during the replication of the DNA. But now, let's see how is the DNA during the cell cycle. What is its form? We said that it has many forms. It can go from that to that and to that, even more dense. Let's see how it goes. No, here. Okay, first, the DNA exists in that form, chromatin. In the G1 phase, the DNA is like a network of chromatin fibers. Chromatin, in this way. We said that the DNA is what? Many large chromatin fibers, like 46 for humans. We said that there is a number. During the S phase, where is it? During the S phase, its DNA is been doubled. So if I have one, let's say I have only one chromatin fiber, it's that, then it is doubled. But is it free? No. The doubled is connected. The two of them are connected in an area and they are identical. So I have this structure, two loosely coiled, they have not coiled even more than the chromatin, loosely coiled chromatin fibers connected in an area of the DNA that is called the centromere. I repeat, the centromere is nothing, not a protein, nothing, it's just the area of the chromatin fiber that the two chromatin touch each other. Okay. And then, during the G2 phase, it starts to coil even more, even more. Because if it's like that, you see, you cannot untangle it. You cannot decide which one takes what. You need to coil it even more. So we get to this stage during mitosis, actually. And this is what a chromosome means. A chromosome, I want to specify that and be very clear because many books get it wrong. And I'm 100% sure about what I'm saying to you because I've discussed it with really good teachers in my university, molecular biologists, histology teachers, and that stuff. So I'm pretty sure about what I'm saying to you. Chromosome exists during mitosis. It's the supercoiled structure, actually no, the supercoiled form of DNA. This supercoiled form is called a chromosome. So you cannot tell me that during the G1, where I have a network of chromatin fibers, you can tell me that the cell has 46 chromosomes. No, that is wrong. During G1, S and G2 chromosomes do not exist. There isn't such thing as chromatosomes. During mitosis, during cell division, where the cellular DNA has coiled to the utmost degree, to the higher degree that it can coil, then at that time we're talking about a chromosome. Okay, it starts during the G2. Where is it? During the G2 to take a chromosome-like appearance, but it is not called a chromosome. I believe I'm clear about that. Okay? Now, Okay, now we saw that until here we do not talk about chromosomes. From here on we talk about chromosomes, but here we only talk about chromosome-like appearance of chromatin. The two, so to repeat what I was saying, until here we do not talk about chromosomes, but like chromosome-like appearance of chromatin. Now, you see we've got two identical chromatin fibers. Because they are more coiled than here, we call them chromatid. Okay, so we've got two chromatids, and because they are identical, they're like brothers and sisters, like exactly. So we call them sister chromatids. Okay, so where is that? Yes, sister. Oh yes, here we are. Two copies of its DNA piece are held together in a structure called the centromere. Each copy is called chromatid, and the two copies together sister chromatids. During this stage, the DNA starts to coil and supercoil. Then we've got the cell division. The cell division is made up in two parts. First, the nucleus divides, and then the cytoplasm divides. The nuclear division is called mitosis, and the cytoplasm division is called cytokinesis. Okay, now during mitosis, which is the nuclear division, we've got the higher level of coiling. Therefore, we now are talking about chromosomes, and they have this appearance. Now let's try to explain this appearance. This is a centromere, and we've got two sister chromatids which are identical. Okay, and these are called arms, the short arm and the long arm. Okay, they are identical next to each other, the secret chromatid, and this is all I wanted to say here. So let's see the stages. Oh, and since the chromosomes have this appearance, they can be seen with the light microscope. Now let's move on. The chromosomes here, mitosis first, nuclear division. Let's see the stages. Okay. First, we've got prophase. What happens at that? First of all, the centrosomes replicate just before the prophase. Okay. The chromosomes start to appear as the chromatin coils up, becoming shorter and thicker, and they are thick enough to become visible when stained. And the centromere with attached kinetochores, we'll see, these are the kinetochores, we will see what that is after we finish with mitosis. Okay. Now, the nuclear envelope starts to disappear. It breaks up into small vesicles which are not visible with the light microscope. The nucleolus disappears because it forms part of several chromosomes. The chromosomes are seen to consist of two identical chromatids. Each chromatin contains one DNA molecule. The centrosomes are starting to move to opposite ends of a nucleus, where they form the poles of the spindle. At the end of prophase, a spindle is formed. And then we got metaphase. This is the spindle, you see, the centrosome at one pole, at each pole of the cell, and these fibers, this structure is called the spindle. You see, each centrosome now reaches a pole. Centrosomes help to organize production of the spindle microtubules. Spindle made from protein microtubules. We've said that before when we were talking about microtubules. We said that they make up the spindle for mitosis. And the chromosomes now line up across the equator of the spindle. And they are attached by the centromeres to the spindle. So what happens here? The centromeres. This is the metaphase plate or equator, and you know the equator there is on the Earth, and this is like that. Okay, we've got the poles and the equator in the middle. And then we've got the anaphase. What happens during that? Chromatids move to opposite poles move to opposite poles, centromeres first, pulled by the microtubules. Let's see that again. No. You see? And each chromosome separates. And then we've got telophase. Chromatids now have reached the poles of the spindle. They will now uncoil again. The centrosome, the nucleolus starts reforming. The nuclear envelope starts to reform as well. Okay, and then we've got cytokinesis, which is what? The cytoplasmic division. It begins during mitosis, sometimes anaphase, sometimes telophase. During telophase, we've got what's seen here, a cleavage furrow. That is called the cleavage furrow. And it moves, and we've got two cells. And so by the end of mitosis, cytokinesis happens, and we've got two separate cells. Now, I want to talk to you about a little bit about the plant cells. In plant cell, a cell plate is forms actually during telophase as vesicles from the Golgi body coalesce, that means unite, down the middle of the cell. Daughter plant cells do not separate from each other. A new cell wall forms, and a sticky middle lamella cements, that means holds together, okay, adjacent cells together. Alright, that's it about. Let's move on now. There are some checkpoints of the cell cycle. I've talked to you about those. I don't think you should know something about these, but I want you to know that they exist. There are points that the cell says, should I keep up what has happened until now? Okay. What is kinetochores? Let me show you this thing. First of all, let's talk about the centrosomes, the centromeres, and the centrioles. The centromere is needed for the separation of chromatids. It is visible as a constriction and is the site of attachment of spindle microtubules. Now, each metaphase chromosome has two kinetochores. These are the kinetochores at its centromere, one on each chromatid. They are made of protein molecules which bind specifically to the DNA in the centromere and also bind to microtubules. Bundles of microtubules called spindle fibers extend from the kinetochores to the pole of the spindle during mitosis. Construction of kinetochores begins before nuclear division starts, during the S phase, and they are lost again afterwards. The microtubules attached to a given kinetochore pull the kinetochore with the rest of its chromatid, dragging behind towards the pole. We said that happens during anaphase. Okay. The poles of the spindle are where the centrosomes are located, one at each pole. As we said earlier in an earlier video, the centrosome is an organelle found in animal cells that acts as mTOC, which is microtubule organizing center for construction of the spindle. Each centrosome consists of a pair of centrioles surrounded by a large number of proteins. It is these proteins that control production of the microtubules, not the centrioles. Plant mitosis occurs without centrosomes. Okay. Now, what is the biological significance of mitosis? I'm going to talk a lot right now, so I don't know. I want you to know just these, but I will talk about them just so you get what each of these are. So, first of all, growth. The two daughter cells formed have the same numbers of chromosomes as the parent cell and are genetically identical. Great. This allows growth of multicellular organisms from unicellular zygotes. Growth may occur over the entire body, as in animals, or be confined to certain regions. Okay, replacement of cells and repair of tissues. This is possible during mitosis, followed by cell division. Cells are constantly dying and being replaced by identical cells. In the human body, for example, cell replacement is particularly rapid in the skin and in the lining of the gut. Some animals are able to regenerate whole parts of the body. For example, starfish can regenerate new arms. Then we got asexual reproduction. Mitosis is the basis of asexual reproduction, the production of new individual of a species by a single parent organism. The offspring are genetically identical to the parents. Asexual reproduction can take many forms. For a unicellular organism such as amoeba, cell division inevitably results in reproduction. For multicellular organisms, new individuals may be produced which bud off from the parent in various ways. Budding is particularly common in plants. It is most commonly a form of vegetative propagation, in which a bud on part of the stem simply grows a new plant. The new plant eventually becomes detached from the parent and lives independently. The bud may be part of the stem of an overwintering structure, such as a bulb or tuber. The ability to generate whole organisms from single cells or small groups of cells is important in biotechnology and genetic engineering. This is the basis of cloning. And last but not least, we've got immune response, which is some cells of the immune system, like the B and T lymphocytes, they have special information in them, and they need to be stayed intact, but they will die eventually. So they depend on meiosis in order for their cloning. Okay. Now, what are telomeres? There are structures. The ends of chromosomes are sealed by structures called telomeres. Okay, these are made of DNA with short base sequences that are repeated many times. In telomeres, one strand of the DNA is rich in the base G, guanine, and the other strand is rich in the complementary base C, cytosine. Their main function is to ensure that when DNA is replicated, the ends of the molecule are included in the replication and not left out. You see, this is why the copying enzyme cannot run to the end of the DNA and complete its replications. It stops a little short of the end. When we will talk about replication, if we talk, you will understand this better. Let me see the specification for a second. Yes, we will talk about the replication of the DNA. Okay. If part of the DNA is not copied, that piece of information is lost. At each subsequent division, another small section of information will be lost. Eventually, the loss of vital genes would result in cell death. The solution is to make the DNA a bit longer by adding some more bases. They have no useful information but allow the copying of genes to complete copying the meaningful DNA. As long as extra bases are added during each cell cycle, no vital information will be lost, and the enzyme that performs this role is called telomerase. The extra DNA it adds is the telomere. The main function is therefore to prevent loss of genes during cell division to allow continued replication of a cell. You see, with each division, some cells, their telomeres get a little shorter until the vital DNA is no longer protected. This could be one of the mechanisms of aging by which we grow old and die. This of course suggests that by somehow preventing the loss of telomeres, we might be able to slow down or even prevent the process of aging. This is all for today, guys. I was talking about the cell, but I have covered some part for the reproduction and inheritance. I covered from the part the cell as the basis of life, the cell cycle, cell division, mitosis, not meiosis yet, and the chromosomes. And from the reproduction and inheritance, I have covered the molecular genetics, structure of DNA. That's all. Okay, and eukaryotic chromosome structure. That's all. Hit that subscribe button if you want to be updated whenever I upload a video. And also, if you want, please comment on the videos from which books of biology you are studying. Okay, I would like to know and have a discussion all together which books are good to study the biology for the IMAT. Thank you so much, guys. Bye bye.